Hi, everybody. Welcome to class. Hi, everyone. Welcome. Hello. Hi, folks. Michael, Aaron, Shelly, Kyle, Lynn, Hallie. Work period. No, come on, work period. You can see the schedule. Ishal, good afternoon. Cheryl, hi. K.E., hi. Lynn, hi. Milena, hi. I saw the eye now. I get it. I get it now. I didn't see the eye there before, but I see it now. Wait, no, we have a lab. Yeah, of course. Diana, you know the schedule. Come on now, Diana. Come on now. Hallie, you too. You know the schedule. Come on now. Don't frowny face me. We got 2 more lessons after this for this unit. And then Friday, we got a bit of a cool period. And then all next week, you basically get work periods. Your wishes come true, everybody. I wish I didn't. All right, welcome, folks. How's it going? Let me just start getting the attendance ready and rolling. We lost someone. Who dropped my class? How? I don't actually think I'll be able to know. Well, yes, I could probably compare lists, but someone dropped my class. I now have 29 kids in this class, and I had 30 before. Now I got to figure out who it is. Oh, I could probably check with the houses. Oh, maybe not. What a shame. Yeah, this class is so fun. I'm glad you think so, Hallie. I try to make it as fun as humanly possible. Who dropped my class? Huh. Well, I'll figure it out eventually. I think I know, but maybe they're late. Message me, because now I'm curious. I can't see them. You can PM me and tell me who you think it is. Because I can't see who it is. Nope, you're wrong. They're still there. Hey, Monica. All right, folks, welcome. It's 1:42. We'll give everyone another minute or two to kind of get settled. Someone dropped my class, which is perfectly fine. I'm just curious to see who it was. They maybe not are in my house situation. Oh, Rachel, I got to add you to a house eventually. And your course selection is due tomorrow, so good to know, Ishal. Good to know. Yeah, Rachel, I'll get you into house on Friday when we do our next house activity. Don't forget to do course selection. Yeah, I gotta see who is on my list that's not in my, let's go last name. Course selections are a pain. Isn't Friday a half day? Yes, and we're gonna do a house activity on Friday, so that's why I'll get everyone sorted into groups, or I'll get you into your groups, and then I don't know what to take. Biology, obviously. I'm not trying to get older. Joke's on you. You have no control. Yeah, half day. I'll explain more on Thursday what Friday looks like. Obviously biology. Obviously. Yeah, who? No, this is bothering me. I got to figure out who. Okay, before we start, I will figure out who has dropped my class. It's not, okay. Yeah. Oh, Mm-hmm. Adit, Isha, Arpita, Aaron, Sitha, Kirsch, Janine, Michael, Ali. Okay, what the heck? I still have everyone in my class too. What? What's happening? I'm having a brain fart, I think, also. All right, I've given up. I'll figure it out one day. Maybe they unroll. No, there's still, I have 29 students on Classroom. Oh, I'm an idiot. Yeah, never mind. They unenrolled on Classroom too. I was like, I have 29 students in classroom and 29 students. Oh, never mind. I'm not going to figure it out. That's okay. We are missing someone today, though. And I think they're not here yet. Yes. All righty. Let me do attendance real quick. Bam. Max brain power. Go back in time and check. Lynn, if I could do that, I would be a bajillionaire. And I'd probably still be teaching, let's be honest. But I would be teaching from a mansion. Oh, no, I'd probably be teaching from like Cancun or somewhere nice, I guess, maybe. Brazil. That sounds nice. Never been there. Sounds lovely. All righty. Welcome, folks. Welcome to class. I'm going to put our schedule up and we'll take a look at our schedule for today. If you've not accessed this file yet, to take a look at what the plan is, by all means, please do so. We're learning about fungi today. I've linked to pizza. That is my favorite. It is a fungus pizza in some way, shape, or form, a mushroom. There's some chili, there's some basil, there's some good stuff on this pizza. It's my favorite pizza of all time. I strongly encourage you to take a look at it. Strongly encourage you to try it. If it's not your cup of tea, that's fine, I understand. But give it a shot. You never know. You never know. So with today's class, I'm going to talk about fungi as a whole. We'll look at some of the aspects of fungi that are interesting. And then I'm going to talk about our lab that I've assigned to you. And I'll go over some details about that lab after we finish our lesson today. All righty. So let's get this lesson started. Let me put it up on my screen, get zoomed in and everything, the whole 9 yards, get it sorted for you all so that way we can get started. Oh, that is shockingly red. All righty. So with regards to fungi and fungus in general, and with regards to some of the other organisms that we're going to talk, as always, we start off by just kind of going over why it's important to study them and talk about them, and just in general, think about fungi as a whole. And the main reason, of course, may not necessarily be pizza, even though it is, quite frankly, one of the most ubiquitous foods on the planet. In my period 3 class, I talked with a couple of students and they're like, I don't like pizza. And I was like, I bet you do. You're just trying to be cool. And they eventually were like, nah, it's not my favorite thing in the world. And I was like, oh, that's different. So you choose pizza sometimes, but you just don't think it's the best thing ever, which is fine. I'm not expecting people to think it's the best thing ever, but it may be the best food ever. Like, how many things can you have this much variety in when it comes to pizza? Like, there's not that many things. Pizza is like maybe the most, you can have the most variety on pizza. I don't think any other food, and correct me if I'm wrong, is there a food that is as variety laden as pizza? Like burgers, no. Noodles, maybe. Like you could do like different type pasta. Yeah, I was gonna say any type of like noodle or pasta, maybe. That's like similar, I would say, in that regard. Yeah, that's a good one, Sitha. 10 points to your house, Sitha. 10 points to your house. I never thought of pasta. 10 points to your house. House of Stitches, 10 points, Sitha. 10 points. Good thought. I didn't really. Soup. Yeah, I guess there is a lot of different types of soup too, isn't there, Lynn? 10 points to House Greenus. 10 points to House Greenus, Lynn. That's a really good one. I never thought of soup either. Soup is the best. I do love soup. Sandwiches. Are sandwiches as diverse? Yeah, I guess they are. I guess they are, Naman. Yeah, they are. 10 points to House of Chimera, Naman. 10 points to your house. That's a good one. Cake. Now, see, I question that one, Lynn, just because cake is it's a dessert, I would say. Or is it always a dessert? Is cake always a dessert? No to cake. Shelly, you don't like cake? Wow. Wow. Lynn, what do you have to say to this? Ishal, you don't like cake either? Cake isn't always a dessert. This is how I love causing these arguments. I love causing these arguments in. Yeah, I guess cake doesn't always have to be a dessert. What other types of cakes are there then? What could you put on a cake that doesn't make it a dessert? I don't think I've ever had ice cream cake still a dessert. Oh, rice cakes. Oh, that's a good one to hear. Bacon, yeah. But then you could still have like bacon and caramel. Pie is also very diverse, Jalen. Pie is also very diverse. I will give you that one, Jalen. I'd say it's also, here's why. Let's pivot this to to Jalen's statement of pie, because you can have savory pies, you can have sweet pies, you can have dessert pies, you can have dinner pies. 10 points to your house, Jalen. 10 points to House Jamp. That's a great point. Oop. That's a great point. Oh, also there are people here waiting to get in. Oopsies. I've been ignoring. We've been so busy talking about. Oh, no, there isn't. I really don't like sweet things. It makes me feel nauseous. Yeah, that's fair. That's fair. House Jamp, 10 points. 10 points. Yeah, because pie you can have sweet or savory. Cake I'm not quite sold on. I don't know if you can have a savory cake for like dinner. I'm not sold on it. I'm not sold on it. I'm not quite sold on it. Yeah. Anyways, okay, so that nice little interlude of pizza and dessert talk and just general food. Chicken pot pie. Yeah, that fun fact, I'm having chicken pot pie for dinner tonight. Turkey pot pie, but same jam, different toast. Same jam, different toast. Turkey pot pie is what's for dinner tonight. Yeah. Salads too. Oh, salads is a good one too, Ishal. You can have all sorts of different salads. I never thought of salads. Ishal, 10 points to House Serpent Siren. 10 points to you. That's a good one. Salads are another good one. Who's cooking? Me. Gotta get this party started after dinner or after class tonight. Gotta make some chicken pot pie, some biscuits to go along with. It's gonna be great. It's gonna be great. Can't wait. Can't wait. I will. I'll upload them to the classroom. I'll upload it to the class. I'll upload it to the classroom. Turkey pot pie with biscuits. Distract them out of the lab, y'all. Yeah, thanks, Jalen. No, nice try. This is a quick lesson, so there's still going to be plenty of time. And on that note, Jalen, since you so wonderfully reminded us to get back to the lesson and to the class itself, let's start by talking about fungi. So, what are fungi? Well, they are one of the most important, yeah, thanks, Jalen, nice try. They are one of the most important decomposers on the planet, if not the most important decomposer on the planet, because they are essential for what's called nutrient cycling. Nutrient cycling. If you've ever watched The Lion King, either the remake or the original, Mufasa talks about the circle of life. And if he was a biologist, Mufasa would have mentioned that the circle of life needs to consider fungi as well, because fungi are so crucial for taking dead or deteriorating life in general and taking it from that storage that it's been, created as a result of its life, whether it's leaves, trees, animals, what have you. And that fungi returns the nutrients back to the ecosystem, back to the ground, back to the earth, to help that nutrients get back into and allow for other living life forms to make use of it. So the circle of life Mufasa was referring to actually has fungi at central and most important to that circle of life, because they help change the forms of nutrients from living things to non-living things and back into living things again. They're also responsible for human health. I'll talk more about antibiotic creation eventually. But in general, the idea of fungi as a whole are incredibly important for that recycling of nutrients. And one way that I like students to kind of like visualize that and think about it is think about early November. Think about early November, October, November. You're outside, okay? You're outside. What do you notice on the ground in large quantities? What do you notice on the ground in large quantities? Early November, late October-ish. You're out for a walk. Maybe you're out in a park, you're in a forest, what have you. What do you see on the ground in late October, early November? Leaves. Yeah, good one. Leaves. Now that you're there in that thought process, take a big whiff. What does it smell like when that time of year? What does it smell like in that, early November, late October? Okay, wet. Yeah, definitely. Definitely. Woody. Good. Yeah. What else? Does it smell like anything else? Or can anyone contribute to think of how it might smell? Worms. That's a good one. Yeah, fungi. Because around that time, as the leaves have fallen, you'll notice when leaves fall in like October at the beginning of fall, right? They have that beautiful orange. They're kind of crunchy. They're fun to step on. You see that it's beautiful, red, yellow, orange, all of those colors laden, those leaves laden on the ground. But as October rolls on and November rolls on, it turns into that brown gunk, for lack of a better word, and rot. Yeah, and rot, because it's fungi that are taking those leaves, breaking them down further and decaying them further to return those nutrients back to the soil. So then at first snowfall, once it creates that layer of snow, the snow and earth, in between that snow and earth is that decomposing layer of leaves. So when the snow melts, come spring next year, what do you notice? Do you still see a bunch of dead leaves on the ground, or are they gone and they've been decomposed fully and returned back to the earth so that those nutrients can be used again to grow all the beautiful plants and flowers in the spring? So again, it's one of those things where you think about it, but you don't actively think about it. And fungi are very important in that decomposition and that nutrient cycling component. So much so that when you do think about fall and when you do think about spring and that gap in between, those leaves got to go somewhere, and it's fungi that help to do that. So it's quite fascinating to approach it from that perspective. It's part of the reason why I find biology so exciting and interesting, because you can start to think of it in that context. And it maybe not necessarily, but it might change the way you approach or view some things because of how interesting it can be with regards to in general with fungi and any living organism and how they contribute to the general circle of life, if you will. So you'll see I have a couple of experiments that I have going on. I'll show you after we get a little bit further into this lesson with regards to some of the things that I want to share with you over the next couple of days, and hopefully next week or so, hopefully before our unit test, just because it'll be cool to show before your unit test. I'm hopeful that some of these experiments will bear fruit come Friday, but we'll see. Anyways, so let's just move on to kind of talk a little bit more about fungi in the context of what they can be compared to. Because when you think about plants and when you think about fungi, they have a lot of similarities, but they also have a lot of differences. One second, I'm going to sneeze. All right, there we go, much better. So one of those, thank you. Thanks, Lynn, thanks, Tahira. Appreciate it, Ishal, thank you. You're well, thank you, Noma. Thank you. All righty, so the interesting thing with regards to fungi, they are not plants. They have a lot of things in similarity with them, but they are not plants. So I'm going to go over some of their differences before I talk about their similarities. And I just wish I'm going to block that up too. And then I'll talk a little bit about that next steps with regards to how they function. So fungi are what's called sessile decomposers, meaning they're made up out of chitin. They don't have to really move too much. And they make their structures out of a soft, spongy material that is called chitin. They also are non-photosynthetic. That means that they do not have the ability to go through photosynthesis because they lack chloroplasts. So that idea that they don't have chloroplasts allows for them to not be non-photosynthetic. So the fact that they are non-photosynthetic and the fact that they lack chloroplasts also means that they are going to be heterotrophs. So these heterotrophs are going to get food from external sources. And even though they don't move, I'll spend some time talking about the structures that allow for them to get that food. So as I mentioned, they do what's called external digestion. I'll spend a bit more time talking about that after this little chart. And then I'll spend some time also talking about hyphae and mycelium, which are just a fancy way of saying the structures that make up a mushroom or a fungi, sorry. So as I compared it to plants, let's talk a little bit about plants. We'll spend more time talking about them tomorrow when we do our lesson on them, the kingdom plantae. But cell wall for plants is made out of cellulose, which is a completely different structure from chitin. It can be a little bit firmer and a little bit tougher depending on how many layers it has. They go through photosynthesis via their chloroplasts. We learned about that yesterday when we talked about protists. That means that they are autotrophs and make their own food. And their structure is made out of stem, leaves, flowers, and roots. So those are their main differences. Let's take a look at their similarities and just kind of compare the two, together. So they are both eukaryotic multicellular. They are sessile. They don't move in the classical sense like we do to go get our food, or they don't have access to the ability to move to go get food. They both grow in soil. And as I mentioned earlier, they are eukaryotic because they have membrane-bound nucleus and organelles. All right, I'm going to give you a minute or two. I'm just going to finalize my attendance. And then if you have some questions, you can let me know. But I'll give you a minute or two to write some of this down as I send in my attendance, and we'll go from there. Alrighty. Any questions about any of the content that we've looked at so far? What does it say next to cell wall for fungi? Yeah, that says chitin. I'll spell it out for you. That's what it says, chitin. It's a type of protein structure that allows for cell walls to be formed. Yeah, no worries. My writing, it got weird. Those periods or those dots are like the capital or the dots on an I. Anyways, won't get into it. All righty, no questions. Oops. So let's talk a little bit about the idea of external digestion as I finish. Oh, 1 second, I gotta. What is sessile? Yeah, sessile means that it does not move, right? So here, I have my one. Let me get this plant for you. This is my one cactus. I call him Spike. This is Spike. Everyone say hello, Spike. That's okay. No need. Hi, Spike. Yeah, everyone can say hello, Spike. Hello, Spike. Yeah, Spike is a good little cactus. How are you? Spike's pretty good. Look at, he's got some new growth going on over here. He's got tiny little spikes growing here. There's another tiny little spike growing right there. How old are you? Spike is, this is the first plant that I got when I moved out of my home. So Spike is about 5 years old now, 4 years old. Spike is the dragon from My Little Pony. That's funny. Hello, little green bebe. Yeah, look at this little green bebe here. Look at this little green bebe here. So this is Spike. This is Spike. Sessile, meaning, do you think Spike has moved from the soil? Do you think he's going for a walkabout anywhere? Or is he quite happy to be right here getting water and sunlight from me, and that's it? He's not going anywhere. He's happy. He's a happy boy, right? He's not going anywhere. That's what sessile means. He's going to stay here. He's going to maybe grow some new shoots out, but he's not going anywhere. That's what sessile means. Meanwhile, if you have an animal like a cat, for example, or a dog, they are not sessile. They are the opposite of sessile. They're always out and about, moving from place to place. Pot them. I don't think your animals would appreciate that. Does that make sense, though, in terms of sessile or non-sessile? Of course, anytime, Marpitha. Good question. It's a good question. Sometimes people take it for granted that they think they might know what that word means. And again, I never take anything for granted. You got a question, there's no such thing as a bad question or a dumb question in my class. So ask away, folks, ask away. Alrighty, let's spend some time talking about external digestion, because as they are sessile, it allows for them to be able to get their food in many different ways. And one of those ways is through external digestion. So I'll talk a little bit about hyphae and I'll talk a little bit about mycelium in the next page, but those structures allow for them to release digestive chemicals into their environment. And as those chemicals break down things, the nutrients will fall and seep into the soil, and that allows for fungi to absorb that nutrients as a result of it externally digesting it first. Whereas other species need to take that food into their cell or into their cell structures and then break it down and digest it. Fungi do not. What if there's nothing around them? Yeah, so then I'll talk about the reproductive cycle, and then I'll introduce you to these two fellas, because these are grapes. You see the date? I don't know if you can see the date. Hold on, you can see the date, right? February 23rd, grapes, right? We got 3 grapes here. They are exposed to the air. This is why you seal things and put them in your fridge. Suffocation, no. You expose them to the air, right? Because fungi release spores that even if there's no food in their general area, the spores that they release allows for them to send out their spores to get food elsewhere. Same thing. This is my starter. I've put it in a little baggie so it doesn't make a mess. And again, I'm exposing it to the air to let those fungal spores have access to it. And if we're in luck, in a couple of days, maybe a week or two, we'll start to see fungal growth on both of these things. And that's how they get their food. Because even if there's no food around them, different species of fungi release their reproductive things into the world and allows for them to grow on new food. Yeah, if you leave grapes in your locker for a year, fungi, they took hold. They broke those grapes down and they used that nutrients to grow more fungi. Sometimes people forget food in there. All righty. It was a lab, obviously. Yeah. A scientist in the making, a true biologist in the making. That's what I tell everyone. Like, oh, do you just have like rotting and decaying food in your house? No. It's science. It's science. Thank you very much. It's science. Alrighty. So let's talk about some of the different species. Yeah, exactly. Let's talk about some of the different species that exist in with regards to different fungi and the idea that there's more than 100,000 different species of fungi, and they fit into mainly 5 phylums. Now, I have noted here, you do not need to memorize all of these things. Maybe I'll use a different color. You do not have to memorize all of these things, okay? And there's 3 more afterwards. I'm just going to do 2 at a time, but you don't have to memorize them. What is important, however, is that you have to know some of the way that they function and behave, the general idea of them. And that should be a little bit easier than memorizing them. Oh, sorry, my apologies. I haven't shared, I haven't refreshed it. There we go. You should see it now. There it is. Yeah, no worries. Okay, so that's the screen I was talking about. Yeah, sometimes I pause it so I can change some things up and block off some stuff, but I forgot to unpause it. So you should all be able to see it now. So we have, you don't have to memorize all of these. You just have to have a general understanding of some of the key features. So we'll look at the first 2, chytrids or chytridiomycota, and then zygomycetes or zygomycota. These are 2 different types of fungi. Chytrids, the first type, they are the first discovered fungi that have what's called swimming spores. So the spores will land in a lake and it'll travel along the lake. And then once it gets to shore, it will then use that traveling ability, quote unquote, to find new food. And then it will digest that decomposing tissues that it's found on the other side of the lake or other side of the ocean or other side of the river. So they're interesting in that sense. They're also known as what's called saprophytes, and they live on dead or decaying matter. So the example that I used earlier with regards to leaves and trees and animals that have died in nature, saprophytes or chytrids are going to be responsible for breaking that decaying matter down. And another interesting thing is that they are single cellular or multicellular. Sometimes they can exist in a single celled nature. Most of the time they are multicellular. So the next type are zygomyocytes, or known as food molds. This is what we're hoping to have grow on some of these fellows in the next couple of days or weeks. And these food molds are also known as soil fungi because they grow in soil or in some type of medium. And we use a lot of them commercially for the production of antibiotics, production of food, among other things. All right, any questions before I move on to the next 3? Any questions before I move on to the next 3? questions? Do we need to remember 1 and 2? You won't have to know their names. You'll just have to know some of their general structures and functions. And you'll see that the vast majority of them are either decomposers or they form symbiotic relationships. Those are like the big takeaways for it. Yeah, for sure. All right, let's take a look at the next three, glomeromycota or glomeromyocytes, ascomycota or ascomyocytes, and basodiomycota or basodiomyocytes. So these three are in some way, shape, or form either harmful or they form symbiotic relationships. So glomeromycota, they form symbiotic relationships with plants and they help the roots absorb nutrients. For tomorrow's plant lesson, I'll show you some of my plants. One of them has a glomeromycota fungi growing on the bottom, and it helps to have that plant absorb nutrients into its roots that it normally does not have access to. That fungi is breaking down some aspects of the soil, and it allows for that plant to absorb some nutrients. Ascomyocytes are yeasts. Many ascomyocytes, like the yeast that's in this sourdough starter, it can be quite harmful to human beings if we consume them because they cause some serious plant diseases, but as well as they can make us quite sick. And that's interesting to think that yeast, which we use to make bread or make other bread, like whatever, pizza dough, whatever you call it, it can be seen as quite harmful to us. But there's one crucial thing that we do to that product that allows for it to be edible. And what is it that we do that allows for this quite harmful yeast that's growing in here right now? We cook it. Exactly. And when we cook it, it destroys that yeast. And in fact, it turns it into some very pleasant flavors with regards to bread, pizza dough, etc. So that yeasty smell you get if you've ever made bread before, you smelled fresh bread baking, that's the yeast giving off all those aromas that once it's cooked, it turns into this very pleasant and quite tasty microorganism. How did people figure this out, though, in the past? Oh, Tahira. You should look up Connections on YouTube. Connections. It's a very old documentary, but it just talks about things happen by happy accidents. Very many cultures, especially in the early AD times, even in the BC times, they ate bread that was unleavened, right? If you've ever heard of the word unleavened bread, many cultures and even some religions, they still have it as a part of their staple in terms of unleavened bread. A lot of cultures in the Mediterranean, in what's now or what was formerly known as Persia, a lot of their breads are flatbreads and unleavened breads, meaning they don't have yeast, right? They don't have yeast in them, so they're not puffy. They don't have any aeration. They're quite, for lack of a better word, flat, but they serve a function. A lot of unleavened breads in South America as well. When you think of like a taco shell is unleavened bread. It's just made out of corn flour instead of wheat flour, right? There's no yeast in it. It's just flour, corn flour and water, a little salt, maybe some fat, and then you press it flat, and then you grill it, and it's cooked, and you have a taco shell or a tortilla. Those are unleavened breads. And one day, I tell this story to myself. It's probably exactly what happened, but who knows? One day, someone was baking bread, most likely a woman, depending on the cultural area, but most likely a woman, was baking bread. So she mixed the flour and the water together. And I'd like to imagine that something happened to her son or her daughter or her partner, and she had this conversation with someone, and she forgot that she was making bread. And this bread and yeast and water, this flour and water just sat. It sat for days until she realized, holy crap, I forgot. I'm supposed to be making bread. And when she came back to it, there was this pleasant aroma, this sweet, sour aroma that this bread had now started to bubble up because yeast had gotten into it, and this bacteria and yeast had gotten into it and started to break down that flour. And she's like, well, I still got to make bread, so I'm going to make this bread. She threw it in the oven, and what she was expecting was this flat bread to come out, came out puffed and aerated. And so she's like, well, we got to eat something. We can't be wasting flour. And they ate this bread. And all of a sudden, you have a yeasted bread with a sourdough starter that's formed by letting flour and water sit to let natural yeast and natural bacteria grow in it, break down that flour, and allow for something not only way more nutritious, but flavorful, aerated, and overall delicious. Yeah, she's like 5-second rule. No worries, it's good. Seems legit. Yeah, so that's my like interpretation of how it happened. I'm sure it wasn't quite that way, but you do see a lot of cultures pivot from unleavened bread, where it's just flour and water cooked or baked, to a leavened bread where you let that yeast sit, ferment, and break down those flours and release those gases and release those flavors. And then when you bake it, it puffs up significantly more than an unleavened bread. So that transition, that shift, whether it's because this one person that I made up forgot about their bread, or whether it was through scientific exploration, test, retest, I couldn't tell you. All I know is that we now know how to make sourdough starter from flour and water. It lets natural yeast and bacteria grow in it. If you do it correctly enough, you can turn just that flour, water, and some salt into a beautiful loaf of bread that you saw a couple of days ago. So it's quite fascinating. A little bit of a tangent, but no matter. I think it's great to go on these tangents to talk about interesting concepts like this. So anyways, as I was saying, growing in here is that ascomycota, which is harmful to us, that if we cook it, it creates this pleasant flavor and deliciousness in bread. And then lastly, same thing with regards to mushrooms, the Basidiomycota, mushrooms, puffballs, bracket fungi. If you've ever had mushrooms before, chances are it was a basidiomyocytes. These mushrooms are what's called decomposers, so meaning they will break down the things in the area and recycle those nutrients into the nature or into the soil. Bracket fungi usually grow on, I can't remember, what are bracket fungi? I think they grow on trees. Let me just double check. Bracket fungi. Yeah, they grow on live trees or live organisms. And they're a type of fruiting body or fungi. So if you've ever seen a tree that has those fungi growing out of it and it still looks like the tree's alive, those are bracket fungi. Yeah, no worries. I was like, I'm pretty sure they grow in live trees, but let me just double check. But yes, that's exactly what they are. Bracket fungi. And like I said, they also form symbiotic relationships with plants. They help to bring nutrients into those plants in some way, shape, or form. So most of these yeasts, as I'll show you as as I clear up some of this stuff, most of these yeasts or most of these fungi, they're either in some way, shape, or form going to be decaying stuff, whether it's decaying leaves or like saprophytes where it decays things in the area, or it decays food that is going bad, or it forms symbiotic relationships, right? Symbiotic relationships with the plants in and around it. They're either going to be decayers like this, or they're either going to be symbiotic relationships as well. And this one, the basidiomyocytes, they're both decomposers and symbiotic relationships. So it's interesting to consider them in that regard. Alrighty, questions for these 5 different types of fungi before we move on to the characteristics, the physical characteristics. Oh, thank you, Arpitha, for letting me know that you're good. Alrighty. Oh, thank you, everyone else who's using those check marks to let me know. Milana, Sitha. I don't know who the other person was. Someone else had it up there for a bit there. Milana, Arpitha, Sitha, Tahira. Oh, everyone's got it going now. Perfect. Milana, Monica. I think I said Milana like 8 times already. Grace, thank you. Add it. Thank you. Alrighty. Let's move on then if there are no questions. And we can talk a little bit about the physical characteristics, because usually people are like, well, hey, hold on a second. I thought you said heterotrophs need to go get their food from somewhere. How do fungi, which don't move, they're completely sessile, how do they get their food? Well, it's because they have what's called hyphae and mycelium. So hyphae forms this mesh-like spongy filament that gives mushrooms specifically their spongy bounce, and it allows for them to be very flexible. And these hyphae create these thin threads of cytoplasm that are surrounded by plasma membrane, wrapped up in a wall of chitin, that protein structure we talked about, which gives it the cell wall. And this hyphae allows for it to kind of be quite flexible and stretchy. And these hyphae grow in networks called mycelium. So hyphae is the individual, whereas mycelium is the bunch or the bundle of hyphae. And these hyphae can move and grow in directions, just like roots of plants. So they grow out from the mushroom. And this is usually where people are like, oh, they found a single mushroom. That's it. That's all they see. But in fact, their hyphae grow for 20, 30, 40, 50 centimeters under the ground to reach out into all the different directions of that mushroom bulb that you see coming out of the soil. And that allows for it to have access to all different types of decaying substances to break it down externally and absorb those nutrients. So the hyphae form, if we'll see them, again, we'll see them on these guys hopefully first in a couple of days. Hyphae form on those fruits and vegetables and food in general. That fuzz that you see when things start to go moldy, that's hyphae forming on top of that food. And it's associated with the mold that's going to hopefully grow on these grapes one day. They are also responsible for producing the reproductive spores that send those spores out into the world, much like when we talked about, I want to say, let me give credit to where credit is due. Who asked that question about what happens if they don't have food? Who said that? I got to find it. Oh, it was Lynn. Yeah, I was going to say, I wanted to say it was Lynn Q. But yeah, that hyphae that they form, as they form that fuzz, they release their spores out into the world to say, okay, we got food right now. But once this food is gone, we got to still somehow survive and move on somewhere. So they release spores, and those spores travel in the air and land on other food and take their time to develop those fungi hyphae as well. So even though they have food, or even though they may not have access to food, those spores are everywhere in the air, on your table, in soil, in your water. And the only way that they can, wait, our notes say that the mycelium stuff forms the fuzz. It should be hyphae forms the fuzz. Like this part here, hold on. This part here, right? Yeah, it should be hyphae. It should be hyphae. That's a typo. Thank you for pointing that out to here. Yeah, the hyphae forms that fuzz that are responsible for reproductive situations, as well as for breaking down some stuff. The mycelium are the large bundles of hyphae that form, like when you look at a mushroom, if you've ever cooked a mushroom or eaten a mushroom before, those large structures that they form, the mushroom cap, those are bundles of hyphae that are tightly packed together to form that general structure. So those are the two structures with regards to fungi, the mycelium, which is the bundles of hyphae, and hyphae are those tiny filaments that are responsible for reproduction, responsible for digestion, and the whole sort. All right, questions about that before we move on? Because then I want to give you all plenty of time after this last point, or this last note, to ask questions. I also want to give you lots of time to take a look at the lab. So again, thank you once again to those people using the check mark to let me know that you're good. I really do appreciate it. It makes it easy for me to see who gets or who wants more feedback or who wants some more, ask some more questions. And it's nice to just see that you're good and you're with me still. I always worry that some of my students, they sign into the Zoom and then they just don't do anything after that. They're just there. And it sucks. I wish you didn't feel that you could do that or wanted to do that, but I get it. Learning online is a little weird and a little difficult. So I understand why you might just want to head out and play video games or read or do literally anything else. So I get that, but it's nice to see some of you just say, yeah, I'm good. I'm still here. I'm with you, Mr. Q. So I appreciate that. Thank you. And it doesn't take you long to click that, right? I'm under the impression it doesn't take you long to click it. I figured as much. Alrighty. So if there are no questions, we can move on to the next part where we look at reproduction. And I will say this right off the bat, folks. You don't have to know this. You don't have to know this. I didn't know that was the check mark thing. No worries, Alex. It's good that you find out now. We still got a full semester ahead of us, so it's good that you're finding out now if that's if you're learning about that. So yeah, beauty. I'm glad you found it. So you don't have to know this entire process, okay? I'm not going to assess you on it. It's a big thing. I usually do a huge activity to talk about it, but it requires us being in person, so I'm not really going to go into too much detail about it. What you will have to know, however, is these points here and these points down here. So I'll leave them highlighted and we'll talk about them, and then you can write some of it down if you would like. But the key thing here with regards to the reproduction of fungi is that they have incredibly complicated life cycles. They are the only eukaryote that reproduces both sexually and asexually. Most eukaryotes just have a diploid nucleus, but most fungi, have a haploid nucleus, which changes the way that they reproduce compared to other eukaryotes. So they're very different with the way that they reproduce. It's a very complex cycle that has them going from diploid to haploid to dikaryotic, which is a new term that you're going to learn down below. And that complex life cycle and that complex reproduction cycle sets it apart from other eukaryotic organisms, okay? That's the main thing I want you to take away from this. Not the entire cycle, that's a lot. You'll learn about that next year and in first year introductory biology if you take it. And you'll have to, I'm sure, memorize it in some way, shape, or form. That's not what I'm after here, though. I want you to have a general understanding that that's how eukaryotes reproduce. And so some of the terminology that's down below that I've highlighted, diploid, just like you and I and any other eukaryotic, we have two sets of chromosomes in our nucleus, one from mom, one from dad. So one half of a set from mom, one half of a set from dad, that two sets, okay? Two sets of chromosomes. Haploid, which is our sexual cells or our gametes, sperm, egg, what have you, they are what's known as haploid. They only have half the DNA of us. And then dikaryotic is a weird one. It's probably the only time you're going to use this word with regards to fungi. Dikaryotic means that it has two nuclei present. One nuclei has one set of DNA. The other nuclei has another set of DNA. And this entire idea allows for fungi to reproduce in a very funky and weird way. Is diploid not from sexual reproduction? Yes. So sexual reproduction happens. Two haploid cells, a sperm, which is haploid, one from dad, egg, which is haploid, set of DNA from mom. They come together to form a diploid cell. That diploid cell then goes on to become a fetus, which then goes on to become a baby, which then goes on to become you. Dikaryotic, however, instead of those two cells coming together to form one cell with two sets of DNA, they come together and then they don't merge their DNA. They just hang out together and form new cells as a result. That's what dikaryotic is. So it's a weird difference from what we're used to. Again, I'm not going to go into too much detail about it. It's just the general understanding that fungi go through a different way of reproduction when compared to other eukaryotes. All righty. So that does it for the lesson today. I want to pull up your assignment in Classroom because there's a couple of things that I want to talk about with regards to it before I let you work on it for the rest of the period. Oh, I got some questions from people. Oh, just a yay. Just a yay from our Pitha. Alrighty. So are there any questions about the reproductive cycle of fungi? I guess is the last thing I want to say. I'll give you some time to digest it. Again, I appreciate those of you who use that check mark. Thank you so much that you're letting me know that you're good. This was a quicker lesson than yesterday. It's not as exciting, I think, as endosymbiosis, but fungi are pretty fascinating in their own regard. And it allows for us to do some funky experiments where we'll track every day and see if gross things are growing in here. Which reminds me, I got to let this starter dry out. It's too wet right now, but then it makes my office smell like yeast. And as it goes rancid, it's less pleasant, but we'll get there. It's okay. It's part of science, I suppose. Gross smells and science go hand in hand. just par for the course. Wait, can you explain the dikaryotic thing again? Yeah, sure. Here's what I'm going to do. Oh. All righty. Can you see my screen now? Okay, so we have 3 cells here. 3 cells. This is diploid. This is diploid, okay? In diploid, we have 1 nucleus. We have 1 nucleus, and we have 2 sets of DNA. 1 from mom, 1 from dad, okay? We say N is equal to 2. N just referring to the number of chromosomes, okay? N is referring to the number of chromosomes. Oops. In the second cell, we have 1 set of DNA just from mom, and we say N is equal to 1. What is this called when we have just 1 set, just a single set of DNA? Haploid, exactly. We call this haploid. We call this haploid. Okay, so haploid. And then lastly, we have our again, 1 from mom and 1 from dad. But what do you notice here? What do you notice here? If the red is a nuclei, how many nuclei do we have in this second cell or this third cell? We have 2 nuclei, each with 1 single set of DNA. N is still equal to 2 because there's 2 total sets of DNA, but because it's in 2 separate nuclei and they are separate from each other, we call this dikaryotic. And that's the difference between those 3. Cool? Yeah, I know it's way easier to visualize this diagram if you're not quite sure. So I get that. Does that only exist in fungi? As far as we know, Arpitha, yes. However, if you recall back from that video we saw on the first lesson of this class with Dr. Alvarado, where he goes out into the ocean and he finds cool and weird new species, right? You remember that? Yeah, so the one species that he talked about reproduced both sexually and asexually, but it alternated, right? So it's one of those things where fungi is the only thing that we know about, but how many unknown species are there that might reproduce with their dikaryotic or some combination of dikaryotic and diploid and haploid, or none of this? How many species do we just not know about that reproduce in a completely different way from what we think we know? I don't know. As far as I know, this is what we're learning, but this is just from what we figured out so far, right? Just think when you're my age, if you go on to study biology, how much cool stuff you might learn and discover just because we just don't know it yet, right? We just don't know it yet. So as far as we know, yes, but chances are it's definitely not just fungi. Biology is great that way. It's a lot of unknown stuff and a lot of still discovering. Yeah. All righty. Let me close this down and share my new screen with you to talk about the assessment, the lab that I've assigned to you. So lab number 2, classifying organisms. You should see 3 separate files. The Excel or Google Sheet here provided allows for you to have the species generated. So I have my last name here. Let's take, let's see. The first person to give me a X in the participants, I'll use your last name. Who's the first person? Oh, who was it? Ishal. Thank you, Ishal. Oh, what's Ishal Saad? So when I type in Ishal's last name, oops, it gives me 10 species. Those 10 species, you'll then copy and paste into, oops, into this chart. Here we go now. Here we go now. Here we go now. There, into this chart where you type the scientific name, record both names if given. Sometimes the species will have 2 different names. Sometimes they'll have two different names. Put both if you get them. You'll tell me if they are unicellular or multicellular, prokaryote or eukaryote, if they are sessile or not, and if they're heterotroph or not, and then which kingdom they're in. Just a quick Google research should help you to determine that information. I'm not looking for you to cite resources like that dichotomous key assignment. Just a quick little Google. You don't even have to put any citations. Just say yes or no for movement, multicellular, unicellular, so on and so forth, and then the kingdom that they belong to. So you'll get that for all 10 of your species. And then after you finish all 10 classification of those species into their kingdoms, there's a couple of questions, 4 questions for you to answer for your analysis. Okay? Does that make sense? Pretty straightforward, I hope. This is a significantly more low-key assignment or lab compared to your first one, I think. My hope is that it's a bit more straightforward and easy to answer. And as you saw from our first lab quiz, doing the lab makes your lab quiz not easy, but it helps, right? It helps. And that's it. That's it, folks. So we have about 15, 20 minutes left. If you would like to work on this asynchronously, you're more than welcome to work on it. I strongly recommend that you get it started. It shouldn't take you too long. The Google Sheet, once you type in your last name, so I haven't changed anything. If I push enter again, it gives you a new list of 10 creatures, right? So it's okay if it changes. It's not the end of the world. It's really just so you have these 10. It randomly generates it, so you're not all doing the exact same one, because that's boring. And it's more exciting this way. Is the second lab quiz going to be the same length as the first lab quiz? Approximately, yes. Yeah, I like a good random number generator or random species generator. If you click around into it, you can see the key data. I've copy and pasted all sorts of different types of, well, spoiler alerts, it tells you some of it there, but that's fine. It copy and pasted some of the species, and it randomly pulls from each of them. So it's pretty interesting. It's pretty interesting how that works. Excel and functions in general. I'm trying to learn, can we not copy the organism? Yeah, you can. Oh, okay. So if you want to copy it, you got to right click, copy, and then you have to probably open up a new Google Sheet to paste special, right? To paste special, because it should work when you do that though, but it's like quite a lot. You'll probably have to copy it one at a time, but I think you'll survive. Paste special. It works for me though. Is that what are you having trouble copying it, Shelly? It should just copy and paste. Okay. No, Lynn. Shelly was just asking, can we not just copy and paste the species? But yes, you can. It wasn't letting you. Was it just recycling it and creating new species lists for you? Yeah, that's okay. You figured it out though, I think. And I think you will all too. So in worst case scenario, if you got to type it out, it takes 10 seconds of your life. I think you'll survive. So we just find info on the 10 organisms and then answer the analysis questions. This isn't marked, it's just the lab quiz. Correct. I use the submission of the lab and the full completion of it as a complete or incomplete. And I started having this conversation with one of my classes, or one of my students in period 2 class. And she was like, if our mark is crappy at the end of the semester, but we finished all the labs and we did them really well, and we completed every single one of them, everything's done complete, can we use that to bump our mark up? And I said, that's a great question. I'll consider it. So even if you look at this lab and say, I'm never doing this, this is stupid, I'll just review the questions and get it ready for the lab quiz. Worst case scenario, if you want to use it as a means to bump your mark at the end of the semester, I'm open to that conversation when the time comes. Cool. Oh my God, what if our mark isn't crappy? Could we still use it to bump our marks? Probably not. I have a question about the lesson from a while ago. Yeah, shoot. Do you want me to pull up the lesson? You don't have to. It'll be quick, I think. Okay, sure thing. Go for it, Arpitha. It was on the lab quiz too. What does it mean by prokaryote staining those colors in the gram and positive cell wall? Wait, are those even colors? Yeah. So I'll pull up what you're talking about. 1 second, because that's a good question. Where's the quiz? Here we go. I know the answer, but I remember, but I don't know what it means. Yeah. Ooh. Hold on. No, that's not what I wanted to do. Hold on. Let me pull it up. Let me pull it up. I'm just going to pull up the actual bacteria notes. There. Okay, so what it means with regards to that. Yeah, part 1, lesson 5. And for those of you who are okay with this, I'm going to explain this quickly, and then I'll leave you to work for the day. Can I delete the NF54C GCA? Yes, you can. That's fine, Cheryl. You can. You don't have to put that in there. Okay, so gram positive and gram negative. So gram negative means it has a thin protein wall and will stain pink with safranin. So it will stain pink with safranin. Whereas gram positive cells with a thick protein wall will stain purple with crystal violet. And that just has to do with the type of stain, the type of bacteria, and yeah, so how does it stain? Okay, so safranin is able to go through that thin protein wall and stain the entire bacteria pink. It stains the entire bacteria pink because the protein wall is thin, and that stain can go through that cell wall and it gets absorbed by the bacteria and it turns it pink. It's like pink food coloring that's absorbed by the cell. Yes, exactly. Whereas with gram positive, the cell wall is very thick. It will not let safranin enter the cell, so it does not stain the cell. But crystal violet, however, it attaches to the cell wall. And so if it has a very thick cell wall, more of that crystal violet can attach to the cell and it turns it purple. So that's how they stain purple and pink. Hopefully that answers your question. Yeah, no worries. No, please, you never have to apologize for a question like that. In fact, 10 points to your house, Arpitha, for bringing up a question from a couple of days ago and still wanting to see clarification for it. I applaud you on that. 10 points to House Jamf. 10 points to House Jamf. Well done. Well done. Well done. Well done. Well done. No worries. Yeah, if people have questions about stuff from before, I'm more than happy to bring it up and talk about it again. It's fun to kind of take a visit back to the stuff we've learned already, right? Alrighty, folks, it's 2:45, which means there's 10 minutes left of class, which I will let you do asynchronously. If you would like to ask a question, you're more than welcome to email me. I strongly encourage you to start working on your lab activity. Otherwise, folks, we made it through another day. I'll see you all online tomorrow. Bye, everyone. Enjoy your day. Chris, take care. Kyle, Milana, Monica, K.E., Ishal, Setha, Lynn, Tanya, Cheryl. Bye, everyone. Have a good one. See you later, Aaron. Have a good day. Arpitha, I will have fun making turkey pot pie. I'll send pics. All right, add it. Hallie, Janine, questions for me, or you're just kicking around for a bit still? See you later, add it. No worries.